Hi guys, this is the Cambridge paper, Cambridge biology paper 4. So I have selected this question to discuss today's class. First we read the question. Domestic goats are small herbivores, animals that provide milk for human use. Goat's milk is an important source of food for people living in rural China. Xinong, Zanon, and Guenzhong are the names of two varieties of goat common in China. In these breeds, there is genetic variation at nucleotide position 5752 of a gene coding for growth factor. At this position, there is either cytosine or guanine nucleotide. Some individuals are homozygous for the allele containing C at this position, capital C, capital C. Some are homozygous for the allele containing G, that is capital G, capital G, and some are heterozygous, simple capital C, capital G. Table 2.1 compares the mean milk yield of the first milk producing period, first lactation, and the next milk producing period called second lactation for Xenon, Sanan goats of each genotype. So genotype at position 5752. So CC, CG, and GNG. First lactation, this gives 679, 622, 616. The highest is this, and then this is the second highest, and that's the lowest. So second lactation, this increased compared to the first lactation, and this increased compared to the first lactation, and this increased compared to the first lactation. Again, all these, this is the highest, and this is the second highest, and that's the lowest in this group. Okay, variation in phenotypic characteristics such as milk yield is caused by contribution of genetic and environmental factors. Goats also show variation in milk yield between the first lactation and second lactation. Suggest. Okay, suggest means you have to find valid information from this, and also you have to add your information to justify the questions with reasons whether the variation in milk yield between the first lactation and the second lactation are shown is genetic or environment. Right, now given a phenotypic characteristics, for example here, the milk yield. So this is equivalent to genotype plus environmental factors. As we know, we have learned this one. There are two types of variation. One is continuous variation, other one is discontinuous variation. So when you talk about the discontinuous variation, discontinuous variations, which is characterized by this kind of a data distribution. For example, human height. So we can find short, we can find extremely tall, but majority are in the middle range. So this kind of discontinuous variations, phenotypes decided by the genotype and the environment. Let's assume that if there is no impact from the environment, then phenotype is 100% decided by the genotype, right? When there's no environment from the environment, the phenotype that is the physical, the appearance or specific character is decided by the gene expression. In this case, if it is happen like that, it's highly unlikely to change this phenotype. But when the environmental factors add, this can be changed. Now, if you look at the first lactation, it is 679. Second lactation, 940. So if you look all these goats, first lactation is less, but second lactation is high. That means something change from the environmental side. So this is, first you have to say, this is environmental. Not genetic, it's environmental. And how you justify now? You can say the first lactation, milk yield is less, but all these genotype, second lactation, milk yield is higher, right? So first is less, but when it comes to second lactation, this increased. This is due to environmental factors such as nutrition, and the maturity of the animal, and then the maturity of the udders. So udders are mammary glands, that mean glands which produce milk. So these are collectively called udders now. And then nutrition, change of nutrition, maturity of the animals and the udders. We can say these are the environmental factors which changed. All right? Fine. So then we go to the next part of this question. The variation at position 5752 of the gene coding for growth factor is due to a substitution mutation from G to C. With reference to table 2.1, describe the importance of substitution from G to C. Right. So if this is a G, this is replaced by cytosine. So we call this kind of mutation is substitution mutation. So when we look at the previous table, we had a C and C, C and G, and G and G. So we have seen highest yield came from homozygous C phenotype. This has the highest, and this is the second, and this is the lowest. So we can say now this change make this genotype, and this genotype has the highest milk yield. So then question asking now, describe the importance of the substitution from G to C. So we can say this substitution increased milk yield. So that's a beneficial mutation. So this causes a higher amount of food for the people in this region where they had this China, where they raised these goats. So they had the more food. So this mutation is a beneficial mutation, right? Okay, next part of the question. In a population of 268 Xenon Saanen goats, the frequency C is 0.30, frequency G of allele is 0.70. The Hardy-Weinberg principle can be used to predict the number of goats with capital C, capital C, capital C, capital G, and capital G, capital G. For example, the number of goats with capital G can be predicted to be 131. Use the Hardy-Weinberg principle to predict the number of goats with genotype capital C, and capital C, capital G. Okay, let's see if we use this Hardy-Weinberg equilibrium. So this capital simple Q squared, this represent number of goats with its recessive form, and this one represent number of goats with its dominant homozygous form, and this represent heterozygous genotype. So you can always write like this: total number, and then the recessive form. So actually here we don't see recessive form, but we know the capital G and capital G is the lowest one, milk yield. So we can use capital G number of goats here. And here, total number. Then this is capital C as the highest yield is given, and this one total heterozygous number of animals. Right, now so P and Q equal 1. This also we know the dominant and recessive one frequency, when you add, it's always 1. So what are the numbers given to us? The total number is given, which is 268. And also, the number of goats with capital G is 131. This also given. And then what else given? Frequency of capital C and frequency of capital G. So now what we did, we just trying to get these numbers into one place. Now what we're going to do, so the C, this one is given, the frequency is given 0.30. So then P squared is 0.30 into 0.30. So this could be a, let me calculate, should be 0.09, 0.09. So P squared is 0.09. And then if you multiply by this with the total number 268, (09) multiplied by 268. So I get 24. And this one Q, is 0.70. So this one 0.70, 0.70. So if I multiply this, I get 0.7 multiplied by 0.7, 0.49 multiplied by 268. So it's 131. So 131, sorry, it's a 0.7 into a 0.7. Yeah, that's 131. But you know, 131 is, this one is already calculated here. So we need to find this one now. So this one is 2 into 0.3 into 0.7 into 268. So let me calculate that one. 0.3 multiplied by 0.7, 268, 113. Rounded up, it comes 113. So this one is 113. This one is 24. Okay, we come to the last part of the question. Total number of goat 268, and then Guanzhong is 440, and we can find number of genotype and then allele frequencies. A close match between your predicted figure in B1 and the actual number in table 2.2 would mean that the Xenon population in Hardy-Weinberg equilibrium. So we found these are here 24 and 113 and 131 was given in the previous, and we calculated this one and this one. State the name of a statistical test that could be used to find out whether or not the Xenon population is in Hardy-Weinberg equilibrium. Okay, we have actually learned during the theory, if we have exact values, so-called standard values, and then we have after experiment we have a predicted value, so observed value, and our predicted value. For example, in dihybrid cross 9, 3, 3, 1, that is the ratio we get in a dihybrid cross. But if you have some values different to that, let's say this is 90, this is 30, 30, and this is 10, but we get something like a 84, 27, 30, 37, and maybe 10. So now this value and this value, are they same or they are statistically different? We can answer that question by performing a chi-square test. So this is answer is chi-square test. Okay, observe and the predicted. The predicted number of goats with each genotype in the Guangdong population, according to the Hardy-Weinberg Principle, are 16, 135, 289. So it is 16, 135, 289. Here, here, and here. So here it's a 47. Here it's a 69 and 324. With reference to table 2.2, describe the evidence that shows that this Guangdong population is not in Hardy-Weinberg equilibrium, excuse me, and suggest a reason for that. Okay, so it's a 3 marks question. We cannot perform chi-square test at the moment for this case, but we can actually check the values again. And the given values 16, 135, 289. So without performing the chi-square test, we can see this is quite higher. It's more than double. This also close to double. And this one also reduced to a higher level. So these values, then we can say, without performing the chi-square test, so this is higher than the predicted. So predicted number of goats with each genotype in the Guangdong population, according to Hardy-Weinberg, is 16. So this increased. This is very higher, right? And then this one is reduced, and this one is 289 is also reduced. So we can say this is these are the things actually we can say, no? So we can perform chi-square test with reference to describe the evidence that shows that the Guangdong population is not in Hardy-Weinberg equilibrium. So we going to say it's not in Hardy-Weinberg equilibrium. If it is in Hardy-Weinberg equilibrium, these values should come very close to this value comparison. Reason is Hardy-Weinberg equilibrium we define based on the several conditions. For example, no mutation, then no natural selection, population should be large, there is no migrations, and there's no genetic drift. We have listed all these things. So if it is not in Hardy-Weinberg equilibrium mean, one of these reasons are not, conditions are not adjusted. So if it's not adjusted, then the population is not in Hardy-Weinberg equilibrium. Okay, so we discussed one question. If you learned something from this video, please make us one like to me. I will post more discussion in the next coming series of Cambridge paper 5, paper 1, and paper 2.